back to my channel. So in today's class, we are going to be talking about cell division. So we all know that cell is divided into two, mitosis and meiosis. Cell division is divided into mitosis and meiosis. But before we go into mitosis proper, I would like us to discuss a bit about cell. Cell is the basic and functional unit of life, and they come together to perform a specific function. The body cells is divided into two, the somatic cells and the germ cells. So what are the somatic cells? Somatic cells are responsible for growth, development, repair of worn-out tissue. So the fact that you grow, you keep on growing every day, the somatic cells are responsible. So while the germ cells are responsible for the production of gametes, and they tend to be seen in reproductive organs. So for somatic cells, we say that mitosis occurs in somatic cells. Mitosis is the division of a parent cell to form two daughter cells that look exactly like each other and that of the parent nuclei in terms of its genetic component. So mitosis occurs in somatic cells. This is done in order for one to perform growth, for cells to divide to their former or similar cells, to carry out growth and development of tissues, repair of tissues. So somatic cells are cells that are responsible for development and growth. So in somatic cells, we have a diploid number of chromosomes. That means there are about 46 chromosomes present in somatic cells. So if a parent cell having 46 number of chromosomes divides to two daughter cells, the two daughter cells will have 46 number of chromosomes each. So that's what we need to know. First of all, the somatic cells are responsible for growth and development. Secondly, mitosis occurs in somatic cells. And thirdly, is that there are 46 number of chromosomes present in somatic cells. So a division of a parent cell means that there will be equal number of chromosomes in that of the daughter cell and that of the parent nuclei. So we come to germ cells. What are germ cells? Germ cells occur in reproductive organs and they are responsible for the production of gametes. Now, unlike somatic cells, meiosis occurs in germ cells. This is to help produce the male and the female gametes. And then the third one is that the chromosome numbers are haploid in the germ cells. This is because when the female gamete has its own 23 number of chromosomes and the male has its own 23 number of chromosomes, they come together to form a zygote that now has the complete number of chromosomes found in the normal human, which is 46 chromosomes. So with this little idea, we can now enter mitosis fully. So what is mitosis? Like I said earlier, mitosis is the division of a parent nuclei into two daughter cells that look exactly like themselves and that of the parent nuclei in terms of their genetic component. So the mitosis has several stages, and the mitosis begins in prophase. And these are the stages of mitosis: the interphase, the prophase, the metaphase, anaphase, and telophase. But mitosis begins proper in prophase. But before then, let's discuss interphase. What is interphase? Interphase is a period of two mitotic division, and in interphase, the chromosomes appear to be thread-like, and then the DNA component in the chromosome tends to duplicate. So I'm going to represent it in a diagram. So this is it. This is our cell. This is our nuclear membrane, and then here you see the chromosomes like threads to be scattered within the nucleus, just as you can see. So what you see here is the cell which we are talking about. Then this is our nuclear membrane. Then this is our chromosomes, but these chromosomes appear like threads, so they are called chromatin threads. Chromatin threads. So as you can see, these chromosomes appear to be long, so they are called long chromatin threads. So they appear to be very long, as you can see. So Interphase explains that it is a period of two mitotic division. It's a period where the chromosomes appear as thread-like units, and they are called chromatin threads for that reason. And also the presence of a nuclear membrane, and also lastly is that the DNA components within the chromosomes duplicate in interphase. So So while we've gotten the knowledge of interphase, let's move to prophase. This is interphase. So now in prophase, what happens in prophase? Just like I said in interphase, the chromatin threads appear long and thin, but here in prophase they become shorter, and then there is presence of a centromere. Chromosomes then form subunits known as chromatids, which are held at the middle by a centromere. So I'm going to present it. You can see here. So as you can see, these are the chromatids. This is our nuclear membrane. So this is our centromere. But there's one thing you need to understand in prophase. In prophase, the chromatids are not clearly seen. Like they are not clearly easily distinguished. So the prophase cannot really say they are chromatids, but with the help of a microscope, scientists have been able to say that chromatids are present in the prophase stage, but not properly distinguished. Now is in another stage known as prometaphase. Prometaphase. In prometaphase, those chromatids I told you that are not clearly seen becomes very clear. They are easily distinguished. So I'm going to represent it. So this is what you see. These are their represented. So you can see the chromatids easily seen. They are well distinguished. So I forgot. Do you have your centrum at this point? So as I earlier explained that in interphase the chromosomes appear like threads. Then in prophase, these threads become shorter, condensed, and are held at the middle by centromere. So they form subunit, chromosomes form subunits known as chromatids. So these chromatids are held at the middle by a centromere. But these chromatids are not well distinguished in the prophase stage. It's when you now come to prometaphase that you see the chromatids easily distinguished. So you can still see our nuclear membrane present here. Then the next we're going for is our metaphase. So what happens in metaphase? In metaphase, these are our chromosomes that form some units which are chromatids. They tend to align in the equatorial plane, and then they tend to be attached to microtubules and spindle fibers for them to be able to align at that plane, and they are seen at the both ends of the centrum. So I'm going to represent it. So you can see. So here is our centrum. So as presented here, you can see these chromosomes to be arranged in an equatorial plane, and so you could also see that each of these chromosomes are lined from one pole of the centrum to the other pole of the centrum. So this is one thing you should know. These are our spindle fibers, and these chromosomes tend to be attached to microtubules and spindle fibers around them. So we move to anaphase. So anaphase. So what happens in anaphase? What happens in anaphase? In anaphase, the striking thing that happens in anaphase is that the centromere splits, and daughter chromosomes form and tend to move to the opposite poles. So this is what you see. Also, spindle fibers are attached to the centromere split as well, helping to carry the daughter chromosomes to the opposite side. The daughter chromosomes also appear V-shaped. So I'm going to represent it. This is what you see in Anaphase. Let me make it. This is what you see. So you could see our center is here. Remember, our center is at each side. The center is here. The center is also here. So you could see the splitting of these chromosomes at the point of the centromere. So that splitting causes daughter cells. These are now daughter cells, daughter nuclei. So it causes daughter cells to move to the other side. So with the help of the spindle fibers are also split away from the centromere. They push them to the opposite side. So you can see them move to the opposite side. Then the next we're going into is telophase. What happens in the telophase? So in telophase, there's a presence of or occurrence of cleavage fluid beneath the epithelial fluid. So this cleavage fluid, what does it do? It tends to bring about division of the cytoplasm to now form the two daughter cells. So there's this cleavage. This cleavage deepens to separate the daughter nuclei, and in it there's also presence of polar nuclear body. So I will represent it. So, this is the presentation of the telophase. Like I said earlier, this is the cleavage furrow. This furrow will tend to deepen, and when it deepens, this daughter nuclei will separate. So, it's with the help of cytokinesis. Cytokinesis is just the division of cytoplasm. So, this cleavage will tend to bring about a separation of these daughter cells for them to become separate. So you can see the nuclear membrane present in each side, and you can see the daughter nuclei positioned in the opposite sides, and then you can see the nuclear membrane on both sides. This is the nuclear membrane. This is the nuclear membrane. Okay, so the nucleolus also reappears. So that's what occurs here. Presence of a cleavage fluid, which you can see, which deepens to separate the daughter nuclei. So this is what we have lastly. This is what you have via the help of cytokinesis. This is what you have. See, after telophase, cytokinesis happened, I will see the two daughter nuclei looking exactly the same. So you could see that the chromosomes begin to uncoil. Just you could see that this almost resembles interphase because it's trying to go back to begin the process again. So after telophase, you get the separation of the cytoplasm has occurred, the two daughter cells are seen, and you can say mitosis has occurred because two daughter cells were formed, the two daughter nuclei are formed. So you should know in all, like I said earlier, that mitosis is the division of the parent cell to form two daughter nuclei that look exactly like themselves and that of the parent nuclei in terms of their genetic components. So this is the process I've shown you. and I will recap that the body is made up of several cells, and these cells are somatic cells and germ cells. So somatic cells are responsible for growth and development, and they are made up of different number of chromosomes. This is because when the parent cell divides, it still transfers everything it has in terms of genetic component, and also mitosis occurs in somatic cell. So this whole thing I just described is occurring in somatic cells in your body, allowing for growth, development, repair of organ tissues, and all of that. Then germ cells, like I said, occurs in the reproductive organ as responsible for the male and female gametes. Secondly, meiosis occurs in germ cells, and lastly, this one has haploid number of chromosomes, unlike mitosis, because the female gamete needs to bring its own chromosome and that of the parents. That's 23, forming 46, which is a normal amount of chromosomes are supposed to be seen in a normal human. So that's all for that, and I'll be recapping on the phases. Interphase, remember that the chromosomes appear slender, they appear like thin threads, so for that reason they are called chromatin threads. Interphase, like I said, their chromosomes appear as threads, so they are called chromatin threads for that reason, and they appear slender. Then another striking thing that happens is that the DNA component duplicates. So secondly, the prophase, which I explained here, now the chromosomes present, they form subunits known as chromatids. So these subunits are held at the nuclei by centromere, and you can see that these threads tend to shorten, unlike the first one, which tend to be shortened and get condensed and thicker. So we move to prometaphase. In prometaphase, just like I said, the striking thing about here is that the chromatids are not easily distinguished. But when you come to prometaphase, you can see the chromatids very, very well. So in metaphase, what do you see? You see these chromosomes aligned or arranged in the equatorial plane on both end poles of the centromes. So that's what you could see here. Then in anaphase, you could see the splitting of the centromere, releasing the daughter cells or daughter nuclei in the opposite centrum or the opposite poles of the centrum, which is represented here. Then next to telophase, which I did not write, next to telophase, in telophase, the daughter nuclei, they move to the opposite sides, and nuclear membrane begin to form in both sides, or forms in both sides. So you could see the nuclear membrane here and also here. Then you could also see the cleavage furrow. This cleavage furrow allows for the separation or division of the cytoplasm. So this furrow deepens, and when it deepens, it tends to separate the cytoplasm into two daughter cells, just as you can see. I forgot to represent it. This is called cleavage. So the essence of this is to bring about or help cytokinesis, division of the cytoplasm. So you can see the two daughter cells out here. That's all for today's class. Thank you for watching. Do well to hit the subscribe button as well as the notification button to keep you updated. Bye for now.